And that's the kind of research I'm interested in, is incorporating a lot of diversity from across the Americas and different germplasm groups to try to make new and exciting corns that have different unique traits. So yes, I'm Sherry Flint Garcia. I work for the U.S. Department of Agriculture Ag Research Service, and I'm a maize geneticist. My technical title is research geneticist, but I'm all corn all the time. And so here we have hybrid corn that is just used as a border. And then next to us on the other side, which you'll see plenty of here, we have a ton of like the real corn, the interesting corn that has a lot of variability. And that's the kind of research I'm interested in, is incorporating a lot of diversity from across the Americas and different germplasm groups to try to make new and exciting corns that have different unique traits. So we have two different sections of our demonstration plots here. One of them is what we call the chromosome walk. Corn has 10 chromosomes in the haploid set. Just like human beings, we have one set from mom and one set from dad. And so when we talk about chromosomes, corn has 10 chromosomes coming from each of the parents. And in the chromosome walk, we planted a long row representing each of the 10 chromosomes. And historically, these chromosomes have been broken up into what we call bins. This was long before they had the structure of DNA and all the intricacies of genetics worked out. And so the bins of the chromosomes were kind of handy ways of telling where along the chromosome you were and how various mutations and phenotypes and things occurred and where they kind of live along the chromosomes. And so we'll be looking at the chromosome walk. We have a lot of really cool things to see in there. And then the other side is more about the history and the story of where corn came from and the kinds of research that happened here at South Farm at the university, both with USDA ARS and with University of Missouri researchers. We do have application in our minds at all times. So we kind of struggle with this as public researchers in maize because the seed industry has really dominated the seed production and the seeds to farmers for growing corn. We struggle with what exactly our role is. It's quite different than selfing species like soybeans and wheat, where breeders in different universities can still release varieties that are useful to farmers. And so we really generate a lot of foundational information about the crop, about the genetics, about all the different aspects of the physiology, the entomology, the insects, plant pathology, things like that, to try to help the industry make better materials. Having said that, there are some regions of there's some parts of corn that are neglected by industry, and I think that's the cool area of research, and I'll be talking a lot about that in a little while. My recent effort has been working on developing food corn. When you think about food corn, you think of sweet corn and popcorn, but there's a lot more other corn that we eat: tortillas, grits, hominy, you name it. And these are not a target of breeding by industry, and so it's kind of up to us in the public sector rather than industry to maybe pick up some of these orphan traits in our really high quality crops like corn that people have been eating corn across the Americas for thousands of years, and they have so much variety, but that's not really available to us when we think about the U.S. corn crop. And so in general, I'm a diversity person in terms of maize diversity. There's thousands and thousands of kinds of corn from across the Americas that are available to us for research and for breeding, and a lot of it's been eaten for thousands of years, and it's really cool, and we should be exploring all the cool things that corn has to offer.